little bit definition study of the genetics. Now, genetics is a study of inheritance and variation, and the term genetics was first used by W. Bateson in 1905. And all of you heard of Gregor Johann Mendel. He's called the father of genetics. He was a monk from Austria, and by him or through him we know what is actually genetics. And remember, it was done in the 19th century, so it was done very earlier, and people did not have any concept of genetics at that point of time. And there are two things, which is one is known as hereditary. Hereditary is what you get from your parents, and variation is the characteristics difference by which progeny differs from the parents. So if you look at yourself in the mirror, do you have all the characteristics that your parents have? No. You have variations in the characters, variations in the attitude, variations in actually what you do. And this cause of variation is due to sexual reproduction. If there were no sexual reproduction, then what would happen? You would have the same characteristics just as in reproduction in bacteria, that is by binary fission, or by just you, like in the fungus, there is reproduction, asexual reproduction, and you won't have any variation in the character, and you would have looked the same as one of your parents. Now, branches of genetics involve cytogenetics, molecular genetics, biochemical genetics, cancer genetics, immunogenetics, developmental genetics, behavioral genetics, and population genetics. So we will not go into the details of much of it, but I would like you to get some information about cancer genetics, and another is population genetics. And thirdly, there will be a description about the genetic disorders. We have already discussed it in the class, but that was a verbal discussion. So we must know what types of genetic disorders are there, what might be the cause of it, and how do we know of it. So genetic information, now we come to different definitions of the genetic information. Gene is the basic unit of genetic information. Genes determine the inherited characters. Yes. So your genes determine what you have inherited, what you have got from your from the environment, or what you have got by evolution. It is not basically determined by the gene. Then comes genome. Genome is a collection of the genetic information. So it is the array of genes. Then comes the chromosome, who are the storage units of the genes. So you have a gene, so that carries the genetic information. Then you have the genome, which is a collection of genetic information, and chromosomes actually stores the genes. And DNA, you have known that DNA is a nucleic acid that contains the genetic instructions specifying the biological development of all cellular forms of life. So DNA is a nucleic acid that contains what? It contains the genetic instructions, so it actually contains the gene, and it specifies the biological development of all cellular forms of life. So this is the chromosome, that is the gene, and that is the DNA. The chromosome has two portions. Actually, we are not much involved with the chromosomes. Chromosomes, we have two portions. One is the locus. Locus is where the gene is present, and allele. Allele is where there is a variant form of a gene. So allele actually is, the gene has two variants. Suppose So they are known as alleles, and the alleles are present at a particular locus. Have you got it? So locus 1, possible alleles are A1 and A2, and locus 2, possible alleles are B1, B2, B3. The alleles complement each other. Then you go to what are genotypes, what are phenotypes. So genotypes is what is present in the gene, and phenotypes are what is actually the expression of the genotype. So your hair color, your weight, your presence or absence of a disease, and genotypes are there 2 genes at each locus, and there you have the alleles, and this constitutes the individual genotype at the locus. So phenotype tells you how you look like, what have you inherited from your parents, and everything of that sort. So you see the genotypes here. These are the genotypes, and the phenotypes look like this. They have black eyes, then you have the white eyes and the black eyes again. So a genotype will determine what the phenotype will be. So the dominant allele in the genotype is actually what is represented, whereas the recessive allele is not represented if the dominant allele is present. But if you have 2 types of recessive alleles, then you can have a recessive allele representation. So dominant allele is expressed even if it is paired with the recessive alleles, as I have told you, and a recessive allele is only visible when paired with another recessive allele. So suppose both of your parents have a recessive allele of blue eyes. So what happens if after reproduction has been a combination of these 2 recessive alleles, then you will be having blue eyes. But if one of your parent has a black eye as a dominant feature and another parent has a blue eye as a recessive feature, the dominant feature, that is the black eye, will prevail, and there will be no expression of the blue eye. So this is one of the structures of the chromosome. It's a double-stranded DNA molecule packaged by histones and scaffold proteins. As I have told you earlier, that the DNA should be protected throughout its lifetime, and even when it's transferred to the progeny, it shall be protected throughout. So how is it protected? The DNA is being packaged into chromosomes, and these chromosomes are actually packaged into small units by the help of the histone proteins, which are basic proteins, and other scaffold proteins, so that whenever you want to have an access to DNA, you have to cross all these barriers. Otherwise, you do not have access to the DNA. Even with all this sort of packaging and preventive measures, the DNA gets mutated. So you can see that what is the effectiveness of these mutating agents. Crossing all these barriers, they can mutate the DNA. So the DNA should be protected, totally protected, and it's protected with the help of this DNA packaged into by histones and scaffold proteins forming a nucleosome. When we will go into the hierarchical structure of the DNA, we will go into the structures of the nucleosome and how it can be formed. But there are 4 histone proteins, and these histone proteins are all basic proteins. You must be knowing that the DNA is negatively charged because it is known as nucleic acids. So since it is known as nucleic acid, it is known as it's negatively charged, and these basic proteins has a very high affinity for this nucleic acids, and they go packaging around these nucleic acids. So genetic concepts are in an individual inherits 23 chromosomes from father and 23 from mother. So how much it is? 46 chromosome, 23 parental, 23 maternal. So each maternal and paternal pair represent homologous chromosomes called homologs. So when we will go back to meiosis, you will see that In meiosis you have homologous chromosomes. Yes, because there only you have a chiasma or crossing over taking place. So chromosomal composition found in most female human cells, 46 chromosomes. Chromosomal composition found in more human gamete, 23 chromosomes. So out of this, 23 chromosomes goes into the human gamete, and 23 chromosomes come from the male human cells and gives you a 46 chromosome total. So homologous chromosomes have characteristics which are, they share the same centromere position. You can just see they share overall size and contain identical genes attached at matching positions. So these are homologous chromosomes. See that the gene for color is present at matching positions at the loci. Then you have the centromere position is more or less the same, and you see GG is actually the dominant part and GG is the recessive part. Here again WW, which is a gene for shape, is the dominant part, and WW is the homologous chromosomes, where their small w is represent the recessive characters. So gene is a sequence of DNA which is transcribed into RNA. With the help of it can be transcribed into rRNA, tRNA or mRNA, and locus, the position of a chromosome on a particular DNA sequence, which I have shown you earlier. So the position of the chromosome of a particular gene sequence, and you at the loci, two positions that they're known as the loci where alleles are present. So if the alleles are identical, they are known as homozygous. If the alleles can be different, they are known as heterozygous. And if only one allele is present, then it is known as hemizygous, case in males for genes on X and Y chromosomes. And DNA is mutable. So this is a known fact, DNA is mutable. Most of the cancers which are being caused are by this DNA mutation, and a variation in DNA sequence or locus is called an allele, which we have discussed for previously shown you the picture. And diploid organisms contains two alleles of each locus. So that is why with the DNA is packaged, DNA is mutable, but this mutation can be rectified by certain enzymes which are capable of recognizing the mutation and then cleaving it off. But some mutations they actually do not go through this checking process, and they provide us with mutable proteins. And this mutable proteins, where some amino acids may be missing, some may be added, some may be different, actually leads to a chaos in the metabolism. And it is very, very difficult to find out what has happened to the protein if you do not know what has happened to the DNA. And in our country, genetic mapping is very, very difficult, very, very expensive, so it is not possible. And the person is given medicines which will actually try to not solve the problem, but actually to give the problem a steady condition. So you should be very much aware that we have to protect our DNA. So if you have an allele G versus G, W versus W, at the G locus either G or GL may be present in a given homologue, either one of them, of a homologous pair of chromosomes. So either it will be G, capital G or small g. And here also a capital W or a small w. Now we will go to someone to the Mendelian genetics. Now what is Mendelian genetics? So Mendelian genetics involves two laws of Mendel. So these are known as the, if you can answer, it will be very nice. What are the true laws of Mendel known as? You can write it in the chat box and I'll be able to see it. So it will be very nice if you can write it down. We will go to that later, but first we will be asking yourself that why did Mendel choose peas or peas on sativum? It can be grown in a small area, produces lots of offspring. You can see by the amount of seeds in a pod. Then you can produce pure plants when allowed to self-pollinate. Several generations can be artificially cross-pollinated. Yes, so it can be artificially cross-pollinated. So if you can cross-pollinate, you will get different types of phenotypes and genotypes, but if you can give for a self-pollination, you will have pure plants. So pollen contains sperm produced by the stamen. Ovary contains eggs found inside the flowers. Pollen carries sperm to the eggs for fertilization. And self-fertilization can occur in the same flower. Cross-fertilization can occur between flowers. Mind you, this is a very nice thing that if the pollen grains which contain the sperm, if they fall on a different species of a flower by any of the carriers of the pollen grain, it does not lead to germination. So that is why nature does not support hybrid plants and animals. It always want to support individualistic properties. So this is a very nice thing that you cannot have cross-pollination by allowing your pollen grains to fall on any other species of flowers and then you germinate. And then you have Mendel hand-pollinated flowers using a paintbrush. Use the snip snappers to prevent self-pollination. Covered each flower with a clogged back. He traced threads through several generations. Just imagine at that time of the world where everybody was actually disregarding science. Everybody thought that science has no foothold on a society. Mendel, being a monk, tried so many things with these flowers, tried to study genetics, tried to crossbreed, did such a lot of things. And so first, how did Mendel begin? Mendel produced pure strains by allowing the plants to self-pollinate for several generations. So now we will be having a live session where you can comment or ask me questions, and after that you will have a IMCRP exam followed by an assignment. So this is how I attempt to end today's class. Thank you.